Hello BIO178, welcome to our Conservation Genetics module, which is actually the second to last lecture content module of the semester, if you can believe that. So why are we talking about conservation genetics? I think being in the EEC concentration, or at least as an undergraduate at Sac State, we all already know why this is an important topic, but just to drive home the point and back this up with evidence, many people believe that we are entering what we're calling the sixth mass extinction event. This is some data from a publication that's cited down here. On the x-axis, we have the time interval, so throughout time, from present here on the right back to the 1500s, and the y-axis is extinctions in terms of the percentage of evaluated species. And we have different colors here, different groups of organisms, mammals, birds, vertebrates, and the dotted line down here on the bottom, this is the background level of extinction that we would expect if we didn't have all of these anthropogenic human activities and problems that we ourselves are causing. And so what we can see is that throughout time, the extinction rate has drastically increased. From the 1500s, it was very, very low, and it has steadily continued to increase for all of the groups that are shown here. And so these are extinctions, but what's also really important to note is even species that haven't yet been classified, that's Enzo, I don't know if you can hear that. Even species that haven't yet been classified officially as endangered, their populations are still declining. So in this graph from another paper by the same author, but more recent, we have different groups of organisms on the x-axis, so land vertebrates, mammals, birds, reptiles, and amphibians. The y-axis here is decreasing species. So in other words, out of these land vertebrates, how many of those are decreasing in terms of their population size? And then the colors of the bar indicate whether that species is already considered endangered, which is shown in the rust reddish colored portion of the bar, or whether they're technically still considered low concern. And so what we can see here is that overall, all of this entire bar is representing species that are decreasing in terms of their population. But only a fraction of those are actually considered endangered, and a lot of those species whose populations are declining, technically from a management standpoint, are still considered low concern species. This is especially prominent for the birds that are shown here, where you see the vast majority of those species that are declining are still technically considered low concern. Another really important point to make here is that let's think about what species are actually included on this graph. These are all vertebrates. So a lot of what we talk about in terms of conservation is related to vertebrates because that's where we have the most data, but it's also really important to point out that there are a ton of species like invertebrates that we don't even really have data for to understand whether they're declining, and we don't have a good plan in place for preventing that and conserving them. So in this module, we'll talk about how we can use molecular ecology data and techniques to help prevent all of these species from going extinct and what we can do to help them. So in this module, things you'll be able to do by the end of this module are recommend conservation units based on available molecular data, explain the major risks associated with low genetic diversity, provide examples of this, and use related data to determine whether negative effects are indeed occurring. Identify appropriate individuals for translocation based on genetic data. That's one of the common recommended ways to help management of species that are threatened or endangered. And like we've been doing, choose a scientific paper that could be used to write a proposal related to conservation genetics. So for that main assignment, here is one paper that I found yesterday. I think I used the search terms molecular ecology and conservation genetics. This is a really cool paper that came out just earlier this year in 2020. And they were looking at genome assembly of this particular type of oryx, which is shown here. And what they were looking at was using this type of whole genome sequencing to provide information about its diversity And also to help inform the planned reintroductions, they had to put the species back out into the wild. Because actually, as stated in the title, it's currently extinct in the wild, but they do have captive populations. So one of the things they were trying to do was look at the genetic diversity of those captive populations and see if they were suitable for putting them back out into the wild to try and gauge whether that would be successful or not. So this is one example of the types of things we can do with conservation genetics using molecular ecology data. And of course, why is that important? Again, this all ties back to our GRFP proposals. And I've gotten a few questions from some of you regarding Proposal 1 versus Proposal 2. So remember, at the very end of the semester, we're writing a full proposal containing all components, and we've been building up to that, right? So we already did Proposal 1, and then Proposal 2, we're building on the additional components of the approach and the anticipated results. Originally, I had planned the topic for this Proposal 2 to be related to something we talked about recently in lecture, basically in between local adaptation and the current point in time. However, because I've seen how difficult it is for everyone this semester to juggle everything and work in this virtual environment, I don't honestly feel strongly about that anymore. If you want to keep building on what you wrote for your Proposal 1 and just tack on the approach and anticipated results for that same idea, I'm okay with that. So if you have questions, please let me know, but please know that overall, I'm pretty flexible with this. But overall, this is, again, the end goal of why we're learning these topics in molecular ecology. What we'll be using them for and how we'll be applying them is using this information for proposals.